everyone. We are now going to go over the T cell maturation and how we track the T cell maturation by flow cytometry. So T cell maturation starts in the bone marrow, and we have the progenitor T cells arising from the bone marrow. So in the initial stages, they do not express CD3, although CD3 is the lineage defining or the lineage specific marker for T cells. The progenitor T cells arising in the bone marrow do not have CD3 expression. CD7 is the marker that is consistently expressed throughout the stages of maturation, right from the immature to the most mature stages in a normal T cell. So the progenitor T cells are positive for the immaturity markers like CD34 and DR, and they are positive for CD7. And then they go into the thymus, where they undergo different phases of maturation to become the mature T cells. So once they go to the thymus, they also start expressing CD10 and TDT. Especially when they go to the subcapsular area, they start expressing cytoplasmic CD3, and also they have the expression of CD2 and CD5. So CD2 and CD5 follows CD7, and they begin to be expressed on the surface next. And at this stage, the T cells split into two. One is the gamma delta lineage, which are like very small number of cells, and they express CD7, 3, and 2. They do not express CD5, and they express gamma delta T cell receptors on their surface, and they restrict themselves to certain organs like spleen, mucosa, skin, and in the peripheral blood. And that's why neoplasms are arising from these areas. The T cell neoplasms arising from these areas, like for example, hepatosplenic lymphomas, are gamma delta T cells that have the same phenotype because they do not express CD5 or 8. They just have 3, 2, and 7. Now, the subcapsular thymocyte has another larger subset of cells going through the alpha beta lineage of maturation, and so they enter the cortex of the thymus now. And so initially they are called as double negative, or some people call it triple negative, because they are negative for surface CD3 and CD4 and CD8. So they're negative for all three. So they are called as triple negative. At this stage, they express CD7, 2, and 5. They still are immature, so express immaturity marker TDT. CD34 is expressed only in the very early stages, and CD34 is not expressed in even in the late stages of immature T cells. So only TDT is a better reliable marker for immaturity. And again, here they do not have any T cell gene rearrangements yet, and then they start rearranging their T cell genes, and then once the T cell gene rearrangement is complete, the cytoplasmic CD3 and the TCR complex combine, and they migrate to the surface, and they become positive for cytoplasmic CD3. And there is also a stage where they are double positive for both CD4 and CD8 in the cortex, and then they mature more to become either CD4 positive or CD8 positive as they go to the medulla of the thymus. And from there, they get different roles. And for example, CD8 becomes CD8 positive memory T cell in the peripheral lymphoid tissue, or it could be CD8 positive effector T cell. And then the CD4 becomes as multiple subtypes. T helper 1 that expresses TBX21, or it could become T helper 2 that expresses GATA3. It could become T follicular helper cell in the germinal centers of the lymph nodes, and so they express markers like CD10, BCL6, ICOS, CXAL13, CD57, and PD1. Like if you remember, the neoplasm arising from these cells are the angioimmunoblastic lymphomas, and they do express these markers as well. And then a subset becomes T regulatory cells and express CD25 and FOXP3, and the neoplasm arising from these cells are the adult T leukemia lymphoma, ATLL, which also expresses CD25 and FOXP3, and a subset becomes T helper 17 cell. So these are all the subtypes that eventually mature from the mature CD4 positive T cells. So the bottom line that you have to remember is CD7 is consistently expressed in all the T cells throughout their phases of maturation, and cytoplasmic CD3. CD3 is initially not expressed, and in the initial stages, it is only in the cytoplasm. As the T cell genes rearrange, they come over to the surface. So they are initially um, double positive for 4 and 8, then they eventually mature to become either 4 or 8 positive T cells. So now if you remember TALL arising from the bone marrow, they will not have either 4 or 8. They'll have some surface CD3, but they will not have anything like they only have cytoplasmic CD3, but they will not have any surface CD3. They will just have 7. Whereas when you see in the thymus, they will have CD3 expression, they'll have CD4 expression, 8 expression, like they are double positive, and they have CD2, CD5, and CD7. So that is because the neoplasm is arising at different stages of maturation. They tend to carry forward the phenotype of their normal counterpart at that stage, which is why a T cell lymphoma, lymphoblastic lymphoma in the thymus is often 4-8 double positive, whereas the one that is involving the bone marrow is often 4-8 double negative. Now, when you get a thymic tissue, when you do flow cytometry on a thymic tissue, you can actually trace the maturation of the T cells. So here you can see that the T cells are consistently positive for CD7, but they are negative. They start as being CD34 positive and they lose CD34 positive as they mature. Same for CD2, they are consistently positive because when they enter the thymus, at that stage, they do express CD2 and they are consistently positive in the thymus. So they are positive for CD2 and then they start as CD34 positive and lose CD34. And here, 34 versus 3, you can see that these are initially 34 positive, negative for 3. And then as they mature, they lose their CD34 and start gaining CD3, and mature T cells express the bright surface CD3. And in the thymus, you can initially see the cells are 10 negative and 34 positive, and then they lose 34 and gain CD10, and then at some point they lose CD10 again, and the mature T cells are negative for CD10. CD5 is dimmer in immature T cells, and as they mature, they express the brightest CD5. CD3 is also brighter in the mature T cells. CD7 is actually brighter in the immature T cells, and as they mature, they kind of become a little bit dim. That's what the bright CD3 population actually has a slightly dim CD7 expression. This is again CD10 versus CD3 showing similar pattern. Initially CD10 positive, then they lose CD10, they start acquiring CD3, and then the mature T cells are negative for CD10 and they express bright CD3. So the most important thing that you will see, the plot that you will see in thymus is the CD4 versus 8 plot. It's very nice to see that because they initially start as CD4 8 double positive. Like if you remember the cortical double positive T cells in the thymus, then as they go into the medulla, they mature into one set matures into 4 positive only, and the other set matures into 8 positive only. So you can see that dynamic population of maturation in thymus, whereas if it is a TALL, you won't see this tracking of population going to either being 4 or 8 positive only. They will just be sitting here or here. Sometimes they can be either 4 or 8 positive immature T cells, but mostly either they are both 4-8 double positive or double negative, but you won't see this pattern of a dynamic pattern of maturation. That's what differentiates a thymoma with just some immature cells that are normal, that are maturing, versus a T lymphoblastic lymphoma where everything is immature. So flow really helps a lot in differentiating a mediastinal mass. And there is another population of cells called NK cells. So there's no clear description of exactly at which stage the NK cells start developing, but it is thought that they are arising either from the progenitor T cell stage, prothymocyte stage, or the subcapsular capsular thymocyte stage, one of these three, or maybe from all of these three. So that's why they do not express surface CD3. They have cytoplasmic CD3. They express CD7. They do not have 4. They can have some 8 expression, may or may not be. They do not have CD5. They are positive for only CD2, and they have the NK cell associated markers like CD16 and 56, and they do not have T cell receptors. So that's very important to remember. NK cells do not have T cell receptors. They have killer immunoglobulin like receptors like the KIRs, but they do not have TCRs. So that kind of helps differentiate between LGLs and NK cells when you see, when you want to differentiate between the LGL leukemia and NK cell chronic lymphoproliferative disorder of NK cell. So very important to remember, they do not have surface CD3, no 5, and no TCR. And they express NK cell associated markers 16 and 56. So here you can actually see the different T cell populations. So these are 3 and 5 positive mature T cells, and we see different colors because it includes different subsets, the 4, 8 positive subsets. And then there is this pink population, which is negative for both 3 and 5, which are the NK cells, and NK cells express bright CD7. Most of the T cells are positive for CD7, but you will often see a small subset of CD7 negative T cells in the peripheral blood or in tissues, but those are just reactive. As long as the number is minimal, you don't have to worry about it. But you have to also remember that CD7 is the marker that is often lost in neoplastic T cells of mature T cell lymphomas. So when the number of the CD7 negative T cells become much more, then you have to worry about it. So NK cells have CD2 also. So you see the CD2 and CD7 positive cells here, and 4 and 8 nicely showing the separation of 4 positive and 8 positive mature T cells. And NK cells are negative for CD4. They have this variable CD8 positive. NK cells are positive for CD7. And here you can nicely see the CD4 positive cells that have a small subset of CD7 negative cells, and those are usually reactive. 15, CD16 and 56 is positive on NK cells, negative on the normal T cells. This is 3 and 1656, where 3 is negative on NK cells, whereas 1656 is negative on T cells, so nicely showing the separation. And 10 should be negative on normal T cells and NK cells. If you start seeing 10 expression in T cells, then you should start worrying about angioimmunoblastic T cell lymphoma, or sometimes other T cell lymphomas can also express He10, so you should start worrying about that. So this is about normal T cell and NK cell maturation. Thank you.